This method can help answer key questions in the biochemistry field, such as the proteolytic modification of proteins, as well as the characterization of inhibitors and proteases and peptidases. The main advantage of this technique is that it allows a rapid screening of the proteolytic activity of proteases on peptides representing the cleavage site of a given protein. The use here is to study proteases that activate viral fusion.
The first step in peptide design is to acquire the sequence of the fusion protein of interest from a public database such as NCBI or the Virus Pathogen Database. Choose the protease recognition site preceding the fusion peptide and include 2 to 3 amino acids upstream and downstream of this sequence. When ordering the peptide, modify it with the fluorescence resonance energy transfer, or FRET pair, MCA at the N terminus and DNP at the C terminus. During the assay, MCA is excited and emits light energy that is quenched by DNP as long as the pair is in close vicinity to each other. However, if cleavage occurs, DNP will not be able to quench the emission, which can then be read by the fluorescence plate reader. Resuspend the peptide according to the manufacturer's recommendations by gently pipetting up and down. For example, resuspend in 70% ethanol to a final concentration of 1 millimolar. If the peptide does not resuspend very well by pipetting, place the tube containing the peptide and the solvent in a sonication bath until it is fully resuspended. Dispense 100 microliter aliquots of the peptide into light dampening or resistant tubes to protect the peptide from bleaching.